Antagonistic selection and the evolution of sex-biased gene expression. Virtually every phenotype imaginable differs between males and females in the same species. And ultimately, those differences, those phenotypic differences, trace back to differences in how genes are turned on or off during development. So this is a kind of a plot that's been done for many, many species. I think this one comes from Tribolium, doesn't really much matter. What we're looking at here on the horizontal axis is expression in females. Vertical axis is expression in males. Each dot is the transcript from a gene. What you can see is a lot of the genes, most of the genes are expressed approximately equally in males and females. But there's a set of genes that are expressed more in females. We call those female-biased genes. And there are a set that are expressed more in males. Those are the male-biased genes. So the question is, how do phenotypic differences between males and females depend upon those differences in gene expression? Well, ultimately, sex-biased expression, sex differences in expression, result from sex-antagonistic selection. That's a situation where an allele that's beneficial in males is detrimental to females, or the reverse. So in this situation, I'm graphing gene expression on the horizontal axis. These are two fitness functions. Here's the male fitness function. Here's the female fitness function. In this particular example, the male and female expression is equal. But because selection is favoring a higher expression in males and lower expression in females, there's sex-antagonistic selection favoring a divergence in expression. Now there's a question that's in the literature that's been outstanding for quite a while, which is, what is the relationship between current or ongoing sex-antagonistic selection and sex-biased expression? So if we see genes that are expressed differently in males and females, is that because they've been optimized and they're doing the right thing? Or is there still ongoing selection trying to get differences between them? So here's my little cartoon of sex-biased expression. It's gene expression here, fitness here. In a situation where the male and female fitness functions are identical, and male and female expression is identical, there's neither sex-antagonistic selection nor sex-biased selection. However, if the fitness functions are different, but the sex expression is the same in males and females, we will have sex-antagonistic selection, but not sex-biased expression. Thirdly, if we have differences in the expression, but those differences are optimized within females and within males, we will have sex-biased expression, but no ongoing selection. And I think you know what the fourth category is. Yep, sure enough. If we have some differences between males and females, but they are not perfectly aligned with what selection is favoring within males and females, we will have both sex-antagonistic selection and sex-biased expression. So anyways, I'm just nailing home the point that what you observe in terms of sex-biased expression doesn't tell you what the current contemporary situation is regarding selection. So that's my question for today. Chenny Ching, who is right here when he arrived in our labs as a postdoc, asked the question, how can we measure current or contemporary sex-antagonistic selection, and can we relate that to patterns of sex-biased expression? So we finally stumbled upon, we had deep thoughts and came upon the following logic. It's a fact of Mendelian inheritance that at conception, allele frequencies at autosomal loci are exactly equal in males and females. So, if there are sex differences in mortality, what will that do? Sex antagonistic mortality generates differences in allele frequencies within each generation. Now those are going to be small, right? There's only one generation. It's not accumulating over time, but they should be there. So, we thought what we would do is relate sex differences in allele frequencies to sex-biased gene expression. How are we going to do that? Well, we turn to public databases. This is the famous 1,000 Genomes project, which confusingly has 2,000 individuals in it. Don't know what's with that, but there it is. 26 populations worldwide. And since we're interested in just sex-biased gene expression, we focused on the coding regions. And for those, we have 6 million SNPs on the autosomes. How are we going to use those data to estimate contemporary sex-antagonistic selection? Well, we'll use the familiar FST statistic between males and females. We'll treat males and females as different populations. If the allele frequencies are the same in two sexes, FST is 0. And with increasing differences in allele frequency, FST increases. So that's our measure of contemporary sex-antagonistic selection. How about data for sex-biased expression? Well, there's a data set that came online recently called GTEx. This is RNA-seq data from 18,000 genes. The data that we made use of come from 14 males, its expression in testes, and 8 females where the expression is measured in ovaries. So those are our data. How are we going to look at them? Okay, we're going to look at a couple of plots that have the following format. So let me just walk through how we're going to think about this. I love PowerPoint. Horizontal axis is the degree of sex-biased expression. So -1 means that the gene is expressed only in females. +1 means it's expressed only in males. 0 means it is not sex-biased. It's expressed equally in males and females. Okay, that's the horizontal axis. The vertical axis is FST, this measure of contemporary sex antagonistic selection. We're going to ask, is there a relationship between those two things? So when we run the data, Shazam, we get a pattern called Twin Peaks. Yeah, isn't that cool? Roll the David Lynch theme song. FST is low when genes are expressed only in females. It's low when it's expressed only in males. It's also low when there's no sex bias. The genes that have moderately female biased expression or moderately male biased expression have significantly higher FST. So we've analyzed these data about 14,000 ways, and that's the most conservative p-value that we've got. It's a highly significant pattern. Okay, so there is a clear relationship, but it's a sort of not obvious or not completely obvious how to interpret that pattern. Oh, by the way, before I go further, let me mention, no single gene shows a significant FST after you do false discovery rate correction. And that's because we've got so many genes we're analyzing. But the point is that if you can give up looking for the particular individual genes that are under selection, you can still find interesting patterns. So twin peaks are seen within each of the 26 populations significantly. And furthermore, the repeatability of FST across these populations. But we'd like to know, of course, is it general to other organisms and to other types of selection? So again, humans, it's mortality selection or viability selection. Technology, working with Drosophila in the lab, looked for genes that are under sex antagonistic fecundity selection. Okay, not viability fecundity selection. And what they also measured gene expression. So they have a list of genes that they have about 8% of the genes are under sex antagonistic selection. And what we're going to do with their data now is not look at FST between the sexes, but ask, and sure enough, Twin Peaks season 2. So here's the horizontal axis, that's the sex bias. Vertical axis is the fraction of low center sex antagonistic selection. And we get this very striking Twin Peaks pattern once again. Okay, so that's the pattern. How can we possibly explain that pattern? So Jenny and I decided that we would build the simplest possible model we could think of that would relate gene expression to selection. And here's how it goes. Assumption number 1, no selection on a gene when it is not expressed. It seems plausible, right? Second assumption, fitness effects increase linearly with expression. That's the simplest assumption we can make. And thirdly, we're going to assume that allele frequencies are at an equilibrium under sex antagonistic selection. And that's the only way we can make the model go down the road, so we're obligated to make that assumption. So what does this look like graphically? I'm graphing gene expression level on the horizontal axis, selection coefficient on the vertical axis. In this particular example, there's an allele that increases fitness in males, and as expression increases, that fitness effect increases. That same allele decreases female fitness. At equilibrium, the selection coefficient on females must balance the selection coefficient on males, and the amount of sex bias is the difference between the expression. So that's the graphical representation of our assumptions. You do some algebra, and what you get is the following. You make the predictions that FST will be 0, when the gene is expressed only in females or only in males. And secondly, for weakly sex-biased expression, you get something looking like that. Critically, that is the delta is our symbol for the amount of sex bias, and it increases quadratically as you move away from 0. So that's the algebra. If you want to see it graphically, here's what it looks like. This is our canonical graph. We've got sex bias expression here, FST here. We're saying that there is no sex, when there's complete sex bias, FST is 0, expressed only in females. Expressed only in males is also 0. And then when it's weakly sex bias, we get this little smiley face. Okay, so that's what the model predicts. And then you do a very sophisticated interpolation, otherwise known as connecting the dots. And you get something that looks like that. Now, that should look a little bit familiar if you haven't fallen asleep already, because qualitatively, yes, that does match the pattern that you see in the data. So this is certainly no proof that the model is correct, but what we're saying is a very, very simple conceptualization of linking expression to selection qualitatively gives us the pattern that we see in the data. Wow, how am I doing on time, by the way?
Got 11:20.
Ah, cool. All right. So, couple of conclusions. And ongoing sex-specific selection is acting on genes with intermediate sex-biased expression. So we do feel that when we see sex-biased expression, that is not evidence that everything is cool and all the sex antagonism has been resolved. For some set of genes, it is ongoing. And secondly, the war between the sexes is more than a metaphor. What we're actually seeing, we believe, in the data is the result of sex-specific mortality on males and females in humans, in our own species, caused by sex antagonism. Thanks very much. Oh, one more thing. We have a job at the University of Texas. Don't leave. Sorry, I meant to mention that. There's the job ad. Please see me if you're interested. Yes?
So why would expression differences in the gonads be related to survival?
Very good question. The question is, why would selection act on expression differences in gonads? I don't know. As a matter of fact, I don't probably think it does. It might be, the expression of gonads is presumably correlated with expression in other tissues and at other times in life. So we cannot say we know what the target of selection is, but we can say we wouldn't see a non-random significant pattern if what we're measuring was uncorrelated with the actual target of selection. There's got to be a connection there somehow. We don't know what it is, and it's a very good question.
Mark, have you broken the data down by sex chromosomes and autosomes?
No, we can't do this trick on sex chromosomes, and that's because sex antagonistic selection will accumulate allele frequency differences across generations. And so we can't do this trick of using it to measure contemporary within a generation selection. So we stuck just with autosomes for that reason. Very good, thank you.